Now we're gonna continue with Nartash from AMBL, and we're gonna be asking your questions to her. I hope it'll be a great time for everyone. So I'd like to start with the first one. Well, it says some studies show that intermediate delay the process of aging. During this, one keeps doing calorie restriction all the time.
Alperen, we can't hear you. If you let me, I'll ask the question until your connection gets better. Our audience's first question is, they said some studies show that the intermediate fasting and calorie restriction delay the process of aging. If one keeps doing calorie restriction all the time, will that reduce its positive effect on aging? What do you think?
Okay, so before maybe answering, I should say that this is a very hot topic in the area, and I'm not actively working on this exact topic, so I may not be very up to date. But still, what I can say is, at least in model organisms, people tried dietary restriction with different durations and also with different age of onset. Like they tried dietary restriction for over life course or after sexual maturation, or for certain periods of times. They still showed positive effects on lifespan. The only potential issue is it's not very possible to assess all phenotypic outcomes in model organisms. So there might still be some detrimental effects, but I should again remind that these dietary restriction experiments, they are all dietary restriction without malnutrition. So they are still getting the food they need to grow and function. So far, no, there's no detrimental effect.
Thank you so much for your answer. Alperen, do you want to try it again for the second question? Alperen?
Can you hear me now?
Yes, clearly.
Okay. Well, the second one is the second one is about depletion in longevity with the selection repression that results from mutation accumulation theory. Isn't it more beneficial to observe the opposite of this data? Like, I didn't quite get the question, but that's what's asked. So the other thing is, shouldn't the life expectancy increase with the selection repression? Like, I think what the person is trying to ask is, if the selection pressure is too high, then it should reduce the effect of longevity.
Okay, so maybe I wasn't clear about that during the presentations. So the idea is that if let's say there's a mutation that has an effect in late life, let's say it increases the risk of mortalities, like, I don't know, cardiovascular disease risk variant. And what happens is, basically, because it doesn't have any effect during the early life, it would still be persistent in the population, and they can accumulate through time because they don't have any effect on survival. So over time, these mutations, these variants accumulate in the population, and because they have detrimental effect on lifespan, they basically just increase mortality, and your lifespan gets shorter and shorter. That's why there's a inverse relationship, like when the selection pressure is lower, then over time, over generational time, you see decrease in lifespan, basically.
Thank you very much.
We have a related question. In the section Evolutionary Theories of Aging, you mentioned the mutation accumulation theory. Could you tell us more about that theory, like how scientists view that theory today? And are there any points where the theory fails to explain a certain phenomenon? If so, what are they? Are there any current studies about those subjects?
Okay, so it's a very broad question. What I can say is, so obviously what it predicts is any variant or phenotype that's related to increased mortality would occur late in life. but obviously there are certain characteristics that may start to show its effect before the selection pressure starts to drop. One can still argue on the balance between source allocation for maintenance and genomic maintenance and growth can still be functioning there, and so in a way what is left after what is not explained by mutation accumulation theory seems like can be explained by either disposable soma or something related to antagonistic pleiotropy. But I'm not very aware of any examples, to be honest, any concrete examples that cannot be explained.
Thank you.
Now I'm moving on with the other question. Do you think understanding the hallmarks of aging can help to stop aging process?
So essentially, actually, it's kind of circular, because the way hallmarks of aging are defined is people just compiled a list of mutations that increase lifespan in model organisms, and then they just allocated them to different biological processes. So obviously, it helps us to understand the regulation of longevity. And assuming aging process is inherently coupled to regulation of longevity, yes, it would help us to understand aging process as well. But obviously there can be differences, like aging rate might differ, but without an effect on longevity, these might be actually missed out. I don't know. Yeah.
Thank you.
Thanks. I'm moving on with the next question. They asked, in a controlled environment, in laboratories, for example, heterogeneity is again observed. How is it tested?
So when we say heterogeneity, I mostly mean molecular studies. Basically, you can either measure the gene activity or certain molecular marks on the genome, like epigenetic marks. What they measure is whether different individuals have differential genetic activity or these epigenetic marks. If in a way, so what we observe, at least in this human data, is people have very similar gene activity, gene expression during their before their adulthood. So which may mean that these genes are tightly regulated. This regulation seems to be disrupted, and then you see this heterogeneity, this variance between people, and it's more or less the same with the model organisms as well.
Thank you. And here's my other question. Actually, I'm addressing the other question. It asks if you've been able to discover a substantial gene or mRNA studying transcription. If so, what's the meaning of it to your study?
So it's asking if I detected any particular gene with. To be honest, no, because, well, so our study mainly concerned with the genome-wide effect of this heterogeneity, for example, the one that I explained at least. And my other studies generally focus much on finding potential drug targets. So again, because I'm a bioinformatician, I generally tend to find these genome-wide signals, and then I collaborate with wet lab scientists to test those. So in one specific example, we tested, for example, a drug targeting HSP90. And we showed that inhibiting HSP90, which is a heat shock protein, increased lifespan in at least C. elegans. Who knows what will happen in humans, but so basically, I think finding these individual genes help us to understand the process in general, because, as I said, with this all the hallmarks of aging bit. But I think if we assume these evolutionary genetic theories of aging are valid, then seeking these genome-wide signals might be more interesting to understand the aging phenotype in general. But yeah, that's what I'm focusing on. But obviously, these individual genes have very much importance to understand the mechanistic behind lifespan regulation, obviously.
Thanks, Hocam. I think that's all of the questions that we get.
Thank you very
Thank you
much.
for joining us.
Comment section. So I think we'll end the session here.
Okay, thank you.
We would like to again thank you for your presence and answering all questions. Okay, welcome back, everybody. So now we're moving on with Arion Avalos and Turul Giray. We're going to be asking you the questions asked by our audience. So I'm going to start first. The first one is, would the defensiveness of the bees suddenly increase after a major environmental change and a stress factor? And can we expect to see higher defensiveness rates with the offspring of the ones that have been under stress?
Aryan?
I mean, I think you probably would be best to answer that because of the pre to post Maria assessment. So that's maybe a good response to that, I guess. So the short answer is that no, we don't see an increase in defensiveness, maybe at the time, but over time, like they don't. I mean, they don't show any signs of having increased defensiveness, for example, after a hurricane, which is a major stressor. But like I said, Tugrud can probably elaborate a bit more on that.
Yeah, so I mean, first of all, the last portion of your question I didn't catch. What did you say?
Okay, sir, let me repeat it again, if you like.
Sure, please. The last part where you said, for example. Yeah.
The second part of the question is, can we expect to see higher defensiveness rates with the offspring of the ones that have been under stress? If you put these species under stress, so would you expect to see higher defensiveness rates?
Okay, so so basically I agree with Aryan that I guess you need to divide this in different time scales, okay? So there is the evolutionary response, which you may think of in one generation, or you may think of in several generations, and however long the stress will be and whatever else gene flow is occurring, okay? So it's the evolutionary time scale. You can also think of it as a sort of immediate response. So that will be within basically neurobiological time scales, neuroscience time scale of milliseconds to minutes to hours to days, okay? And then you can also think of some sort of ecological time scale. So, within the months following the event. So it really depends on the type of stressor. So if the stressor is a predator, for instance, visiting the colony, attacking, collecting bees, etc., the immediate response will be a defensive response. So the colony that seemed like doing nothing, all of a sudden can turn into a monster that can kill you. This we have experience with Africanized bees in Ernesto Guzman's apiary in Mexico. So we're looking at a hive, and it doesn't look any different from other bees. These were supposed to be killer bees, but they're foraging, minding their own business. So one bee scientist, Greg Hunt, says these don't look any different, and takes a brick and throws it at the hive. Okay. Following that, we are all escaping, scattering, and the animals are stinging. These bees are stinging everybody that is in the neighborhood. Thousands of meters, about a kilometer away. So yeah, that will be the immediate response to that specific stressor, which may be perceived as a predator trying to, open the and gain access to the hive. Now, that kind of abrupt interruption may probably leave no effect whatsoever after 3 days. We know that for 3 days they remain upset. So their brain biochemistry is altered, and you can look at gene expression, and find that a colony that was disturbed 3 days ago is basically just turning into its baseline after that 3 days. But during the 3 days, they are still upset. And that's for European or African SBs equal. But if this stressor is always present in the environment, such as this predator, then you would expect some response that is actually colonies that are better at responding to that predator may end up surviving and reproducing more than the others. Therefore, then over generations, you may end up selecting for a different type of response. In Turkey, Syriaca bees show this response, and first, like in 60s, Rutner had shown that Syriaca bees basically suppress their foraging behavior when there is this wasp that is attacking them in the environment. So that response is not observed in other bees. Mellifera, or other bees don't respond in the same way to the presence of the wasp. And also, we recently have shown that there is this heating behavior, boiling and heating behavior, that also got selected over time. So you could then you're basically altering responsiveness to stimuli and type of response, and that's sort of pretty constant from then on. Okay, and whenever the same stressor shows up, the colonies respond reliably in the same fashion. So basically, the idea is that you should look at this question in the immediate, in the ecological, and in the evolutionary time scale.
And to give a little bit more to fill in sort of the conjunctions there, like, so I mean, that immediate response is a continuous thing, right? So like if you have a predator that is consistently sort of disturbing the colony, right, you do have an initial increase in defensiveness, but then if it continues for long enough, the colony goes through a period of fatigue itself, so that then defensiveness actually goes down. Even though the colony keeps being disturbed. And this is work by Claire Rishkoff in Jim Robinson's lab, where she basically went and disturbed the colony over a prolonged period of time to kind of see how that response sort of elicited, and that's the pattern that we saw. So, but that's the transient sort of immediate response. That it needs to be, it needs to be something that is happening consistently and that it affects the ability of that colony to reproduce in order for you to see it fixed in the population. So if you're talking about defensive populations, they're defensive because they live in an environment that maintains that defensiveness, right? That makes it beneficial for them to be that defensive. And so that's why you have some variation among the populations, even within the same continent, honestly.
Thank you very much. That was a very elaborate answer. And I think we shall move on to the other question.
Can I ask?
Yeah, sure.
I write that and I will read it. We know that African bees are aggressive, very aggressive. Is it possible to reduce the aggressiveness of African bees by crossing with Puerto Rican bees or Syrian bees?
So to clarify a little bit, right? What we know is defensive, right, is the Africanized hybrid. And there are reports, for example, that the source African population, so Scutellata, in its environment in Africa, it actually has a range of defensive-ness as well. There's people that say that they go and they visit the colonies and they're not that bad. There's people that say that go and visit the colony and they're just as bad as the mainland, type of thing. So that defensiveness may be a natural thing in the sense that it just be an expression of we got unlucky and the ones that made it to Brazil were a particularly defensive population, or it could be like a hybridization effect, which there's been suggestions that might be the case. So the fact that they're a hybridized population, so like an admixed population, might have led them, to be a little bit more defensive. But it has to again be maintained in the population because of environmental factors. If not, like it usually, doesn't get selected, and so you end up having a normal distribution, or it gets removed entirely if it affects some other trait, which we know is the case for foraging, for example. And so yeah, I mean, so to say that if you just mix this with this, right, you can lower the defensiveness. That's not always going to come about. And there's examples of that, like in Mexico, for example, they've been trying to lower defensiveness for something like, I mean, it's now probably closer to 30 years or something like that, through both careful sort of breeding programs and then through sort of more general beekeeper efforts of just flooding the area with European honeybees, for example, which that's kind of how both of those have gone. Some have been directed breeding efforts with European honeybees, and others have been sort of like a beekeeper in a valley just floods the valley with European drones and things like that. And both of those systems, they work temporarily in the sense that like they may change that local population, but the moment those progeny go on and get placed somewhere else where there's more access to sort of that Africanized background or that defensive sort of trait, the trait breaks down rather quickly because it's still adaptive to be defensive. So that was the argument that we made in sort of the work on the Puerto Rico population, is that propagule that made it to Puerto Rico from the Africanized population was likely defensive, but it was really some level of environmental factors going, basically determining that defensiveness was not adaptive in Puerto Rico. And so like it disappeared. Whereas in the mainland, it's probably still quite adaptive. And you can see how that's the difference. I mean, Puerto Rico's an island, really the only predator and disturbance that a bee colony might encounter would be humans. So it's, there's not as much need to be defensive. And in fact, it would be to their detriment to be defensive, because then that brings the humans' attention. So that means, more pressure on them to not be. Because then, I mean, there's not much place to go in an island, right? And it could also be like other factors, like Tudulu showed in his previous works, where there's basically environmental criteria that forces the bees to focus more on foraging. And we know that once that happens, sort of defensiveness tends to go down because the two are competitive traits in some ways. And so Tudulu might have more on that as well. So.
Yeah, so Aryan, that was I think very very good, very complete explanation. Just one thing that I would like to add is that there has been experiments where crosses to see what happens just when you cross, and then in the Africanized bee case, this hybrid population that sort of de novo. Because of us present in the Americas, the defensiveness was at the same or close to the same level of Africanized bees, even though you're crossing them with an Italian Ligustica honeybee, which is famous for not being aggressive. So those kind of things happen, and then so it depends on the genetic background. And we are thinking of paternal effects. We are thinking of so there is complications to just being able to accomplish. Although it could be possible, through breeding efforts that are more sort of directed, and even within the same population, things could be accomplished. I mean, this is the premise why. Aryan has a job.
Yeah. Actually, on that note, like we actually have a similar situation. We have a trait in our lab here in Baton Rouge that is for varroa resistance, right? And one of the hardest things that we've had to think about is how to maintain that trait when we give it to beekeepers. Because the moment you put these varroa resistant bees, similar to defensiveness, right? The moment you put them out there in the world, and they go on for a couple of generations, that trait tends to sort of go away. We think it's because maybe there's some it's just the colonies that are not having to worry about that are treated, and as such don't have to worry about varroa, can have their cake and eat it too, right? But the resistance we want, because we don't want to be dependent on chemicals, right? So, I mean, the point there is that answer has not been we just cross them. It's been we cross them, but then we need to figure out a logistical plan to be able to have the beekeepers maintain that cross, right? So it ends up being something like, well, okay, then the beekeepers have to routinely go back and get sort of that original cross into their hands, because it really doesn't work that we just leave them with it, because eventually they'll lose the trait in their local region. And so defensiveness may work something like that, and that seems to be the case with a lot of honeybee traits, honestly. So.
Well, I mean, so the issue is this: when you are doing a selection program, you are focusing on one particular gene, and particular trait, multiple genes. Typically, for a single trait or couple of traits that you're looking at, probably with simple genetic background for whatever you're looking at, with major effects, you may need something like 3,000 colonies as your population base for selection. And that then you can have those two traits go hand in hand in your selected population. But the program, as Ernesto puts it, is a marriage, a long-term marriage. You cannot divorce. As soon as you divorce, you lose what you have selected for because of gene flow. So how does it work in nature? Well, for Puerto Rico example, this island population became different, but remember that's 150,000 population, not 3,000. All individuals on there with no gene flow from outside, considerable gene flow from outside, and that they are subjected to the same selection pressure, continuously, which the selection pressure includes both lack of selection for aggressiveness and increased selection for foraging. So it's evolution that under natural conditions works sort of more robustly than anything we can try to do selecting breeding wise. Okay, so I guess that's the answer.
Well, it maintains it better than we can. I mean, we can isolate a trait. So that's the thing with honeybee breeding, right? So you can actually isolate traits rather quickly compared to other organisms. I mean, and there's plenty of works where that's been shown with regards to broad pages, sort of high and low pollen or pollen hoarding lines. Sorry, the language is not coming through. So for example, there he was able to do that selection independently twice, and within the span of 5 years, which is unheard of in any other organism, he was able to recreate those two lines, right? High royal jelly lines in China seem to be within the same time scale. We know, for example, from collaborators there, that they've been able to breed for that trait for within 10 years. Again, unheard of in terms of breeding programs that usually they take 20-30 years to even begin to see change in the trait, honestly, if you're talking about like cows or chickens, for example. So I mean, this can happen quickly in a breeding program. It's the retention that matters, right? And for the retention, the environment can do a much better job than we can, because we are not there all through the generations all the time applying the same selective pressures. We're just there when we make the breeding choices, right? Unless we stay next to the beekeeper and sort of give him the bees every and even then we're pulling from the same bucket effectively. So that would be the change that's needed, or it's a logistic solution if you want to talk about it in those terms. If you're a beekeeper, for example, interested in obtaining non-defensive population rather than a breeding solution or a crossing solution, let's say.
Thank you for your answer. Can I add something related to this question? Do you have any efforts to increase the response rate of bees to flowers, different species of flowers?
You mean like, for instance, make the bees visit a certain type of crop, for instance, increasing crop pollination efficiency. Yeah, I mean, people, we can do things of that sort, okay? So we can do things of that sort. For instance, there are flowers they like to go. For instance, alfalfa flowers, which has 70% sugar concentration in nectar. So bees like to go there, but they don't pollinate it. They figure out quickly not to trigger the mechanism that will dust them with pollen. So they are sort of nectar robbers, and we can select so that they will not mind or not remember how not to trigger. So we can select for stupid bees that will visit that flower effectively. So so you can play with their preferences or abilities, etc., in selection. So to increase visit rate or pollination efficiency on a particular crop.
On a general sense, I will add that that's part of what we do as well in terms of our breeding lines, but we take a very broad look to it. It's not so much as specific to the flower, so much as, so for example, we have in the US these large migratory beekeeping operations, which span the continent effectively. And so they take, thousands of colonies, usually to California for a specific pollination window in the spring. And then they, then move them off to different states depending on which route they're taking. So our current lines that we have been developing for all of them, really, part of the thing that we do is we actually, once we have the trait isolated, we then take that population, open it up a little bit, like let it breed with more lines that we think are optimal, and then take all that and put them through this ringer, basically through this harsh sort of migratory beekeeping operation. And part of what we measure as doing this is like, well, how much honey and how much pollen, for example, that those colonies, headed by those queens that we've selected are doing when you compare it to a baseline sort of, Italian honeybee. And so part of the effort is in improving that visitation, right? Because if they have enough pollen, that means that they've been pollinating, right? And so it's kind of like a proxy, more general way, but it's a way that we can quickly and on a large scale able to improve that particular characteristic. And so we measure that throughout the season. It's like a longitudinal study every time we do this. So so it's not trivial. It's actually pretty intense, but because there's hundreds of colonies usually at a time and keeping track of multiple of those migratory routes. But usually, hopefully, the end result is going to be bees that are quite good at pollinating or collecting honey, depending on what that particular lineage was desired for.
Yeah, so I mean, in agriculture, one thing is, for instance, almond, which we are increasing the acreage, incessantly, let's say, to the point that, bee and pollination, it's a limit. So we have to take all the bees in the US, almost all of the colonies, to provide pollination services. But in addition, we don't have enough water for these trees either. So it's really not sustainable environment-wise, human existence-wise. So we do that. And to that effect, we also try to come up with these little solutions. One solution being what Aryan was mentioning, to change, as you say, their behavior, right? To have them shift to pollen rather than nectar, because that's what mostly the almond pollination is about. Less nectar, but more for pollen early in the season. February is the pollination date. So if you have colonies that are more inclined to collect pollen, that will be better. So that's one thing that all that group with Greg Hunt, Rob Page, etc., began and has done these studies in California, so there is that. The other thing that I can think of, that will be good. When we were doing this pollination work in crops, the one idea is that you want to get hybrid vigor, which is on the plant side. So So to get hybrid vigor, you need cross-pollination. And that's a terrible, difficult problem with the bees learning very well. As I was telling in my talk, they learn very well how to handle one flower, and they learn all the color, etc. And that's a preference at that point, that they will go for that. So what we want them to quit what they're doing and also try the other flowers. So, for instance, in Walter Farina's research, they were showing for sunflower pollination that most bees just go to one type of flower. So they are really not causing cross-pollination. So one is collecting pollen, the other is collecting nectar. They don't really see each other. What really happens is some contamination when these same individuals come into the colony. Therefore, some pollination occurs. So it will be really nice. You can have some bees, at least some percent of the time, switch their preferences. In Italian bees are notoriously sort of persistent on one thing. And we were telling that as the fact, that's the only thing. So in biology, we used to just, when we study an insect, I mean, this doesn't include me, it sort of predates me, you would say the cockroach or the honeybee, okay? One organism representing the whole species. You know, in your question, there was something like species. No, let's talk about individual populations and individuals and the variation, okay? So that's what evolution is about. We're not really focusing on doing anything for species. So species is genetic construct, but what is being selected are the individuals, okay? And then what evolves maybe is the populations changing. So those concepts are important to keep clear. Now, basically, that idea of unitypical, uniform representation by a single cockroach representing all the cockroaches and any other insect, actually. Because do you know how many species of cockroaches exist in the world? Any of you, including Aryan?
It's gotta be probably. I have a book that actually says, because I read it like 5 years ago, but it's probably something in the millions, maybe. I want to say.
No, species.
Oh, species. Okay.
That's like 3,000.
Yeah.
So, and we only know 2-3 of them, the Americanum.
Yeah, I know that like the ones that are pests are like 1% of like all
Right.
the species of, or 0.01% of all the species of
Yes,
cockroaches.
0.01% are the ones that are associated with us. So, and the only Periplanata americana is probably the only cockroach these guys ever studied, and within that species, probably only one lab population. So it's really not representative of all the behavior that is available out there in nature, okay? So in the case of honeybees, the same thing. We based everything that we were saying on one bee that was a mongrel hybrid, looked like an Italian bee in US, basically, okay? So for the longest time.
In northern US, let's be specific on
Yeah,
that.
That's it. So now I mean, and that's before Africanization, etc.
Yeah.
So now we are starting to barely scratch the surface of this variation. Aykut Kence, this is a nice topic because Aykut Kence is one of the people who first looked at this difference. Within Turkey, you have about 20% of at least subspecies level variation represented. And each of those animals behave differently. And they are changing the textbook understanding of many learning and foraging, etc., related phenomena. So Caucasica, for instance, it is not going to go to the same flower even if you put a gun to its head. It's sort of going with this idea that you cannot have spring with a single flower, like tek çiçekle bahar olmaz. So that's the Turkish saying. So they will visit multiple flowers, even if they are going to a flower that is rewarding. About 25% of the time, they will visit other things that are blossoming around. So that will be a great thing for either sunflower pollination or for any other pollination, cross-pollination needs that you may have. Our one study showed that typically bees can reverse, the flower that they were going, but at some period they only go to one thing, right? So that's called reversal learning, and this is something that is common to us and to the rat, to the cockroach, to the human being. So if you go back in history, that's the way it is described. So if you cannot, for instance, forget what you were eating before and keep looking for that, then you are abnormal according to this paradigm. Okay, so if you may like a lot, like Turkish bread for me was the thing that I couldn't find in US, and I wouldn't accept the spongy Wonder Bread that you can squeeze into like maybe to the size of a fist, the whole loaf, and it just disappears and doesn't taste like anything. Turns into cement probably later. I don't know. So the Turkish bread, on the other hand, as you know, crusty, fluffy, very nice. Now, you should be able to reverse this. I mean, I should be able to reverse this affiliation and say, no, I really like Wonder Bread is the wonderful thing. Put some peanut butter on it and with jelly, it will be great. So I should be able to make that switch, okay? And if I cannot make that switch, then something is wrong in my brain, okay? So, and that's sort of the paradigm with bees too. So if a flower A is blooming and it disappeared, in a few days, flowers are ephemeral, right? Then the bee should be able to find another odor, another color association with another flower and follow that. This is what the Italian bees do, and that's what we think all the bees should be doing. What we found with Syriaca and Caucasica, this was, as a result of research initiated by Aykut Kence, we were examining this foraging traits. And what we found is that Syriaca basically does not give up, sort of like me with the Turkish bread, does not quit the Turkish bread and does not accept the Wonder Bread. Okay, so that's like Syriaca. So it will form an association and not quit that. Whereas Caucasica will eat both. I mean, so we'll quickly switch to the Wonder Bread, and then as soon as maybe goes back to Turkey or finds a bakery that has the bread, we'll go for that bread. Okay, also. So we call that an expanding repertoire. Okay, so it is able to increase the associations forming this bee, whereas the Syriaca seems to be sticking to one thing and not doing the reversal. Both of these are very different from either human beings or the rat or the cockroach or the Italian bee. Okay, so that has to do with all the natural diversity that you're seeing. Italian bees, on the other hand, what they will do is if they are in Turkey, they will eat the Turkish bread, no problem. And if they're in the US, they will eat the Wonder Bread and no problem. Okay, so that's what it should happen. In neuroscience, if you are not able to do this or do it differently, then we start wondering what is happening neurobiology-wise. Okay, so that's a little take on foraging behavior. So that could be something that one can select for, perhaps, depending on your needs.
Another, and just to tie it up, another component is that you actually, so I mean, Tuğrul is touching on, when you talk about predilection for a flower, right? It's not only the bee getting to that flower, it's also the bee being able to successfully obtain the resource from that flower and the colony being able to survive on it, right? So these processes are not just like, can you train some bees to go to this? It's like, great, yes, you can, probably, but at the same time, like if there's something else out there that they prefer, they're more than likely going to go for that as well. So there's that. Sorry, go ahead.
May I ask something? You have said that bees memorize certain tastes and colors and avoid them. Can we trigger this situation with plant grafting? I mean, can we include other bees, including stupid bees, as you call it?
So you mean oh, okay, you mean manipulating the landscape so that you can train the bees to avoid or go for a particular resource is what you're okay. I mean, on a lab scale, we've been able to show that you can do that. So they're capable of learning and forgetting, right? To some degree, as the group mentioned, there's heterogeneity in honeybees, and so certain populations are able to do this more easily than others, right? But on a large scale, I wouldn't know enough to say. I mean, that requires a lot of engineering, right? So, and a lot of, large scale testing. So it's one of those where you really wouldn't be able to per se. I do know that from our own experiences, again, with the migratory routes, these bees grow from Louisiana, where it's relatively heterogeneous in the spring in terms of flower availability, to like California, and there's only almond there, right? I mean, like 4 miles. And so, we're not giving them a choice when we do these sorts of movements, or not, not we, us, but the beekeepers, let's say. Similarly, if you're taking your bees, to another resource, so from California, for example, they go to alfalfa pollination in North Dakota to kind of replenish their energy. For some of them, others go other places in the U.S. But so when they do that, again, we're not giving them a choice. We're planting them in a spot where for miles around there's nothing but alfalfa, right? And I know in between there, they're walking through states that have been completely, what's it called, completely farmed to produce, or structured to produce corn and soybeans. So there's really nothing in between there for them, anyways. So these are the level of large scale engineerings that have already been done for crops, right? And that we're just kind of tossing them into. So our response in our lab has been, well, we need to pick a bee that is going to be able to survive that. Because the what you're recommending is more, can we instead rearrange our agricultural landscape, right? So that we can selectively sort of discourage or encourage. And that's something that's very hard to do, because you can change your plot of land, but there's 30 different farmers usually around you that may or may not be involved in your experiment, right? And so for every correct association that the bee is forming in your plot, there's 100 different incorrect associations is forming in the plot next to yours, right? So I mean, that's the plots adjacent to yours, let's say. So that's the type of thing that if you're talking about that sort of landscape engineering, that doesn't come from the bee, that comes from the farmer, and that comes from sort of the country sort of directing this land.
So there is, at some level, efforts to this. I mean, we do it in agriculture, for instance, even for corn. You would basically plant in a way that you will have two different varieties next to each other, and then you will hire people to remove the pollen producing, start from one and then leave the pollen from only one of the strains. So you would have cross-pollination. So we do go to in these kind of efforts. That one is just mechanical and human aided, and but still you plant them so that there will be pollen transfer between these two lines, where in one you are basically removing the pollen source. So we do that for bee pollination too, when designing orchards, etc. So people do put these different strains. These kind of things happen, for instance, for cherry, breed production. So you want to have cross pollination that increases yield, and as a result, you're trying to place them in ways they will increase chance of mistakes by bees. I mean, not necessarily good association the way you asked, but at least to have them make enough mistakes that there will be pollination. The other thing that people do, for instance, the cherry case, they do collect the pollen from one strain, turn it into dust, and then place it at the entrance of the colonies. So when the bees are exiting to go to the flowers, they end up touching this pollen and affect cross-pollination. So yeah, we are coming up with ingenious ways, sometimes tedious, to use the current behavior of the bee to be able to achieve the pollination need we have. So for instance, in this experiments on if this kind of planting the certain matrix of planting the different strains in an orchard will lead to cross pollination. A few things that we see is that you maybe pollen sources you put every 2 trees or 3 trees, something like that. But when you have a regular interval, it turns out that bees figure out that, so they jump and go to the next one. So there are it is some area that is being studied and that is being used in high yield crops. Okay, so on things that are worth it your effort. Otherwise, like Aryan is telling, it's best to have a bee that's sort of general purpose, and if it is accidental, the pollination, just because bees bump into each other in the colony, well, so be it. You increase the density, you calculate the density of how many colonies to put there. The other thing that you sort of use, for instance, with almonds, etc., you want to cut all the undergrowth, because if not, you will have clovers that are giving very nice sweet nectar, and bees will go to that. So you want to eliminate that. And then the other thing that we use is element of surprise. So you reduce competition is one. The other is to use element of surprise, which is you bring your bees after a certain very low percentage of flowers are seen. So the big bloom will happen immediately. So you bring the bees right before that bloom. So when they are in this new environment, the first thing they see is this thing. So sort of like when you move to a neighborhood, maybe you will shop from the first store that you see there on the corner. And then later you'll figure out that you paid like 5 times for the same stuff, and there are much better quality. But you will learn that over time, right? Not the first day you're gonna, you gotta take the hit. So that's sort of what happens with our pollination efforts with bees. We just take them to the place just as it is needed, and it is a surprise. And then we also have reduced the competition. So yeah, there is a lot of agricultural sort of engineering going on in all this for increasing pollination efficacy.
Yeah, more to your question too, it's like that is innately utilizing the behavior of the organism, right? All of this process that we're discussing, sometimes is on the, again, on the land management side, and sometimes it's on the what we do with the bees and how we bring them in. But all that is training. Like, right, all that is behavioral entrainment. It's what you're doing in some way or another, whether you're not calling it data, you're calling it migratory beekeeping, but it's like that's what you're doing. You're sort of dropping something in the middle of a novel environment, and it has to acquire all these associations, right? And then it has to stay on that long enough, and so you're cutting it off right when the associations are beginning to decline, and they're starting to figure out things. So all these are nuanced experiments that are already being conducted over and over every year. It's just that a lot of them have been shaped by the logistical limitations of what we can do, right? And that's why I was saying that it's usually easier to just deal with what I'm planting here, because that's going to be the pervasive component, as opposed to what, in what way the bee's going to learn, a new method of me arranging what's here already, right? And as the group pointed out with the cherry, sometimes we do that, and sometimes we do that because it's honestly worth it. In terms of economic reasons for both the farmer and the beekeeper. So I will say there's also offsets of this, for example, so where, people are becoming more aware of the need for having sort of a healthy bee population, right? And so sometimes they do things like sacrifice their own crop production in the case of corn, for example, and have these like stretches in between the corn that has just wild planted flowers, and that's to provide forage in a place that is otherwise depauperate of forage. And that's another level of engineering, right? Where you're basically not teaching the bee to learn a new association, but at least you're giving them a reprieve from like the monoculture of corn, which is it's not going to provide them with all the resources they're able to do. And so there's both ways. There's ways in which we cater to the bees, which is what these strips are, and there's ways in which we manipulate their behavior in order for them to cater to us, let's say. So.
Well, thank you for your answers, and I really it's been such a great session, and I think that's all for today because we're running out of time. So thank you again.
Thank you very much. This was
Thank you for the invitation.
Thank you for answers, hocam.
This is fun.
okay. So I guess this is goodbye.
Okay.
Yeah, for now.
All right.
Hello again. We're together with Ozan Bozda for the academic question and answer session. How are you today?
I'm doing fine. How are you?
We're fine. First, I would like to start by asking you, this came from the viewers yesterday. How did you realize that oxygen levels of the atmosphere is correlated with multicellularity?
Oh yes, so it is something that I didn't realize. It's in the literature. So we knew that for the last, I think with much more confidence for the last 20 years. So we have a lot of studies that track the relative change in the oxygen levels. So you just look at the rock record and track the elemental composition like iron 3 versus total iron rate. So it changes over time because it is oxidized. So we knew the timeline, like the first oxygenation event, we knew that with a good confidence that it started at 2.4 billion years ago. And from 600 to 400 million years ago, we know that the second oxygenation takes place. And we already knew that the eukaryotic multicellular life also shows some sort of an overlapping trend from the fossil record. So fossil record, you know that what time period certain rocks come from, so you just overlap this kind of two types of information, and that's how you know it.
Thank you. Then I continue by I think it is the oxygen question. You mentioned that the low concentration of oxygen in the boring billion repressed macroscopic multicellular evolution. What would you think if the oxygen concentration was extremely high in the boring billion? Does that situation increase the rate of evolution, or might there be any adverse side effects of extreme levels of oxygen?
Yeah, that's a good question. Like, we know that the first oxygenation is actually a bad event for life forms, simple microbes, because they're not adapted to a world where oxygen is present, so that it becomes toxic in the beginning, but different life forms quickly adapt to the oxygenation. And the other part of the question is, if there were high levels of oxygen during the boring billion, whether we expect large size to evolve. I think that is a universal assumption that there's always some tendency for the evolution of large multicellular size for different selective reasons, and that should exist during the boring billion as well. So oxygen itself doesn't promote or select for large size, it just permits it. But if there are other selective pressures, and if there is high oxygen during the boring billion, then we expect for sure in some lineages, in only a few lineages, that large multicellular life macroscopic life should evolve. So I think that clarifies the question, right? That should be answering for it.
Thank you.
Okay, may I ask something?
Sure.
In the evolution experiment, the small and intermediate snowflake yeasts are eliminated, and bigger forms are allowed to divide and reproduce.
Yes.
So in nature, shouldn't the small and intermediate ones not be? Where was I? I was reading. So can I start?
Sure. Yeah, sure.
In the evolution experiment, the small and intermediate snowflakes yeasts are eliminated, and bigger forms are allowed to divide and reproduce. So in nature, shouldn't the small and intermediate ones not be eliminated because they are not disadvantageous? And so their number wouldn't be decreased because they don't have such a disadvantage against the bigger ones in oxygenic environment. What is it that makes bigger ones advantageous in oxygenic environments?
Yeah, I think, this is a very useful question because it allows me to explain some of the things that I kind of ignored during my talk, to make the talk, focus on a single narrative structure. But that is, so for the first part of the question, we expect the small and, intermediate sized multicellular forms to be advantageous when there's a selection for them to be, large. So in my experiments, we use this model, developed by Will Radcliffe back when he was doing his postdoc with Mike Travisano. So they came with this artificial model system that we can apply selection for large size, very strong selection for large size. So it is a system that we use in the lab. So it's not a system to recapitulate what happens in the nature. In the nature, there are other agents that select for large multicellularity. And I'm talking about large multicellularity, meaning that transition from single cell microbial life to, simple diffusion limited multicellular life. And for that to be selectively advantageous, for instance, one thing is predation. Escaping from predation is one of the most important selective forces, where you have single cell that is very important predators that are feeding on small microbial life. So to escape from such a selective pressure, you evolve to form simply relatively larger multicellular organisms so that you can escape from predation. The other thing in nature, to form large groups, the selective force is, for instance, dispersal. So, microbes are not actually motile, especially in land environments, so they cannot really freely move around. So when there are scarce resources, the nutrients are limited. What they do is they form these fruiting bodies, these multicellular bodies, so that they pop off unicellular naive offspring that can disperse away from this place where the nutrient is limited. So dispersal is another selective force. The other thing I think very important is the resource sharing, which means that some of the complex, resources, nutrients like starch, that sugar molecules are bound by chains, so they cannot be captured by the cell easily. What cells do, microbes do, is that they produce enzymes which go to the outer surface of the cell where it degrades the sugar so that it can capture the smaller sugar molecules easily. So what happens in that case is that because sugars are also diffusing away from the cell, they cannot really capture all of the benefit, even though they pay the cost to produce the enzyme, like biological cost to produce the enzyme. So when you form a multicellular organism, multicellular group, the diffusing nutrients are also going to go to your nutrients neighbors, going to go to the neighbors in your multicellular body, so that you'll be consuming nutrients much more efficiently, which also shown in lab experiments done by, for instance, Andrew Murray, where they force yeast cells, single cell yeast cells, to grow in nutrients in complex sugar environments where they had to produce, extracellular enzymes, and then in the end they saw that multicellularity evolved. So there are many different, other than this, there are different reasons in nature, selective forces that select for the transition from single cell to simple multicellular life. So we expect those to be present in the boring billion on Earth. So it's not, we shouldn't think of it as a competition. Being small is most of the time advantageous. You are single cell, you are capable of your own reproduction. So you don't, for instance, being a multicellular body is risky in the beginning because you forego your own reproduction for the higher level unit, right? So transition to multicellular life is a tricky business, but in cases where this selection is strong, there's always, universally we've seen that these different selective agents select for multicellular life, and that should be present, as I said, in the boring billion. So I think the thing we do in the lab relies on a very simple system where gravity selects for multicellularity. It is good because you don't, for instance, have to grow things with predators, for instance, to select for multicellular life, which makes experiments much more simpler, easy to track. That is why this gravity-based selection system is useful, but it's not to recapitulate what happens in the nature. So I really like the question because it gave me the chance to explain other agents of, selecting for large multicellular life. I don't know, Enes, if this explain. This is a good answer to the question you asked, but
I think so. I think so.
thanks.
Selim, you can go on.
Okay. There's a question. Okay. You said that there weren't any experiments on other than this on multicellularity because there weren't any suitable model organisms before snowflake yeast. I couldn't see. Could there be any other model organisms that we can test these on in the future? Can we find other model organisms?
Yeah, that's a very nice question. Actually, in our lab, there's a very talented PhD student, Kai Tong. He uses, different unicellular relatives of all animal life, metazoans. So, we have this large group which has a lot of different clades in it, the metazoans, which are, described by their multicellular nature, but they have these unicellular relatives that are living on the planet today. And he is also using those simple relatives of us, unicellular relatives, and growing them under different oxygen concentration to select for large multicellular size, again by applying cell link selection. And I think related to animals, simple multicellular diffusion limited animals that don't have any cell type specification to transport oxygen. I can give an example of placozoans. It is important, these are very simple micron scale multicellular organisms on which you can apply the size-based, if you can apply the size-based selection, and you can grow them under different oxygen concentration, then you can apply it to such other models. It's just, you need to be able to grow them in different oxygen concentrations, so it shouldn't have a detrimental effect on the organism. And secondly, you should be able to see the effects of selection quickly. So you need to work with things that have short generation times. So I can imagine there should be other model systems, and this should definitely be tested with other model systems to tell us that, if you work with the models from different lineages of life, then you can really generalize these results to other simple macroscopic multicellular systems. So yeah.
Thank you.
You're welcome.
Can I ask one more question?
Sure.
While the amount of blue chromosome decreases in the third slide about the recombination clock, the amount of red chromosome increases in the next generation due to recombination. Because all genetic diversity turns red over time, can we say that recombination reduces genetic diversity here?
Yeah, I think this is not really related to my talk. So you mean third slide, right?
Yes.
Okay. So maybe it's a question for a different talk. I'm not sure, but I worked with recombination previously.
Presentation, I think.
Okay.
Maybe it was written wrong. I'm sorry. I'm not sure.
No, it's just I never talked about recombination in the talk. That's why I am puzzled. But if you ask the question again, maybe I can help.
While
maybe it was written wrong because we took them from the messages. Maybe someone copied it wrong. It could be that. Maybe we can ask it again, or I'm not sure.
Selim, you said this was the topic, right? Last question.
I think that's wrong. Yeah, we grouped the questions before we asked them, but I think it was copied wrong.
Maybe. I think, yeah. So recombination, if I understand the question, does recombination decrease the genetic diversity? Is that the question?
Yes.
Yeah, I think yes.
Okay, so I studied recombination previously and speciation. So the thing is, yes, mixing things are whatever the slide says, answering something that I haven't seen, but whatever the slide showed, recombination actually creates new combinations of alleles between individuals in a same gene pool. So that these new combination of novel allele interactions actually create much more novel, phenotypic responses in the populations of individuals. So recombination, because it creates not just additive, changes, it also creates epistatic interactions, which you cannot predict the outcome of, so that you can create novel phenotypes, novel traits with recombination. That is why bringing things from different individuals through recombination in the same, individual is useful in terms of creating genetic diversity. But I can see why they think that mixing things up decreases the, genetic diversity. But the outcome of that mixing things up actually genetically creates novel traits and novel, consequences in organisms. So that's why it is useful for increasing genetic, diversity. I'm not sure if that answers, but
Thank you so much for answering that as well.
Is there any question left?
I think that was the last one. Let me check again. I think, yes, it's the last one.
cool.
And thank you so much for joining us and answering these questions, and also sending your presentation.
Thank you for all the hard work. I mean, I know that it is really hard to organize these events, and I know how much work you guys have put through it. So I really thank you, and thank you for inviting me. It was a lot of fun, and I started watching all the other talks, and they are super enjoyable. So it's going to be a great resource on YouTube. Thank you. Bye.
Bye.
Thank you for your contribution, Hocam.
Thank you, Enes. Thank you so much.
Thank you for that.
Thank you. Creating my chat window just in case you might want to type certain things, or I might need to type. Sure.
Okay.
Hello, everyone.
Hi, Hujam. Welcome.
Thank you for having me.
Thank you for being with us. How are you, first of all?
Very good. It's snowing outside. Perfect weekend. And so after this session, I'm ready to play with my family in the snow. So it's perfect.
Nice. So if you are ready, we can start with our questions.
Of course, yes. Send me the questions.
Yeah. So the first one is from, I think it was from our NS social media account. It is, how does MYB R2 R3 evolve in the first place? And when these 32 transcriptome factors are downregulated, how is the organism affected? So basically, evolution of R2 R3 ways in the first place, and the affection on organism. That's what I get from.
Yes, R2, R3 is a very, very ancient group, a class of transcription factors. So I mentioned that it's all in the plants, but also in animals and in invertebrates. So it's pretty much in all eukaryotes. We can say a pan-eukaryotic transcription factor family. So for that reason, it controls very key, very ancestral processes in a cell. Can you imagine a gene that is also functioning in a plant and in an animal, right? So it has to be very, very ancestral. And because of its ancestral nature, they can diversify in so many different ways because a lot of time passed, right? Since the diversion of plants and animals and other eukaryotes. So in plants, this is a very question because genotype to phenotype connection. It can be very complex. It can be very complex, and it can be ultra complex if it is a transcription factor, because we know that transcription factors are, I don't like the saying like switches, right? Molecular switches or gene switches, but they do serve as a certain, they do play a huge role. They have a larger weight compared to other genes. So the question is, what might the R2, R3 factors doing in lianas? Is that the question when all of them are turned down? Was that the question?
It is not stated in here, since your economical presentation is on lianas, it makes sense.
Yeah, so let me answer that on that part at least. So R2, R3 are transcription factors, but not all transcription factors work exact the same way. So a typical transcription factor is, okay, it goes, binds into the promoter of number of genes, right? And then makes them active. But sometimes it does the opposite. It binds into the promoter and represses them. So first of all, I need to sit down and see what's going on with the upregulation and downregulation nature of a particular transcription factors. And as I said, there's 32 of them, so I need to read a lot of literature there. But let me give you an example. So MYB44, right? MYB44. I know that this is downregulated in liana, and when I look at the literature for that, it controls a stress pathway in plants. So we know the plant leaves have those stomates, like they open and close, right, for gas exchange. So this transcription factor negatively represses, so it represses, it doesn't activate certain abscisic acid, that's the plant hormone, abscisic acid sensitive receptors. So this is very interesting because lianas, if you remember, they have really large vessels, like the picture with all these like xylem and phloems. They're like huge, and so because of that, they need to control their transpiration rate more than other plants. So this might be a way, just one of the transcription factors. So we don't know how all these transcription factors will work together to create a phenotype, but I'm just giving one example, MYB44. So these transcription factors may control really mainstream uh, processes, so that uh, a function that is well established in trees, for example, right? Because their way of life is so standardized in our thinking, right? They have a huge stem and then they have a bunch of leaves on top. But liana is very different. So probably MIB44 is making those tweaks to adjust to the liana way of life. So the other group, for example, is controlling the phyllotaxy. What is phyllotaxy? The arrangement of leaves so that they don't shade each other, right? That's like a helizonic arrangement of leaves, phyllotaxy. And phyllotaxy also uh, uh, guides the shoot of the plant to the growth direction, right? If you're a liana, you need to find that exact direction to reach the canopy. So these types of MIB type transcription factors are controlling certain physiological processes and also the plant architecture. And so altogether, some of them are activators, some of them are repressors. I believe these are coming together, and in concert, the whole is greater than the sum of its part, like this famous saying. So one by one, when you look at them, okay, they control these functions, but when you put them together, so you need to approach like a systems biology approach, let's say. So this is just a transcriptomics layer that I'm looking at, but there's also like proteomics layer or genomics layer or other layers, epigenetic layer. So we need to have a systems approach to understand what's going on in altogether what these MIB transcription factors do. At the moment, I don't know, but when I look at the literature, I was able to at least understand the function of 2 or 3 MIB transcription factors. That's what I'm seeing. Stress control, uh, maybe the desensitization of the stress mechanism, uh, because of architecture is very different in lianas, so they need to control their stress slightly different from the rest of the plants, and also the plant architecture, the phyllotaxy. So anyway, I think that might be a good as short of an answer as possible for a complex question like that. So.
Duru?
Since you mentioned large vessels in your answer, there is a related question. In your presentation, you stated that in almost every vascular family there is a liana member. Is this an evolutionary? I'm sorry, it's the wrong question. Is there any hypothesis about the question of how can lianas have large vessels without suffering from embolization, which you actually mentioned, but
yeah, so that's a long-standing question. We still don't know, because embolism, or some people call it cavitation. So it's basically an air bubble forming inside those tubes, right? Water and nutrient conducting tubes. And from humans, for example, we know that embolism can be fatal. Those divers, for example, deep sea divers, if they come up to the surface really fast, nitrogen bubbles can form in their veins, and you get an embolism, and you can easily die. That's very, something we know as humans. And in plants, by the way, lianas do have cavitation. It's inevitable. If you have a vessel that size, it's inevitable. You will have a cavitation. Probably the question is, what do you do to repair it, right? How do you fix that problem? So lianas must have found a solution, a biological solution to this problem, so that, okay, I will have embolism, but so what? I'll repair it and continue my life. So that's the type of attitude. So what do lianas do to repair this cavitation? And the question is still out there, because some of the vessels are not living. For example, xylem, right? Xylem is a non-living tissue. So how do you fix a problem in a tissue that's not living, right? So you have to interfere it from outside. So xylem parenchyma, for example, the only closest living tissue next to the xylem. So our eyes must turn into that tissue and see what's going on in that tissue that's purportedly repairing the problem. Now, my data set is concerning leaf, so I can't really say what's going on in the stem by looking at processes in the leaves. I can't conclude certain things, but I can't make a definitive statement. So the next, I think, is to get a liana, probably a grape stem, right? And look into the transcriptomes of the stem and see what's going on over there. But on the other hand, there are certain interesting biological discoveries going on. For example, the model plant Arabidopsis, right? Arabidopsis thaliana. They did some grafting experiments and looked at the transcriptomes in the leaves and stems and roots. They found that a huge majority of messenger RNA travels inside the plant. So that's an interesting this discovery. So all that time I was thinking, okay, if I'm looking into the tissue of a leaf, then I should talk about leaf, and I can't make my conclusions on the stem. Maybe that's not true. Maybe I can do conclusions based on stem, but we're scientists. We have to be conservative in our, conclusions. So at the moment, I'm saying that I have to be conservative, and I can only answer leaf problems through a leaf transcriptome, not stem problems from a leaf transcriptome. So let me go back to the original question. What do lianas do to fix this problem, right? Maybe there is an interesting water pumping system that evolved. For example, in plants, we know certain proteins called aquaporins. Aquaporins are the ones that enable water transport from one cell to another. And so maybe they developed a special class of aquaporins, but these aquaporins probably are only expressing themselves in the stem, and I won't be able to detect them in the leaves. So I think next question is to maybe narrow down the research and look at a more specific part of the question. So my suspicion is that lianas have a large class or a large expression in aquaporin class, and they're pumping water probably into the embolized xylem and then fix them. And actually, in my second picture after that big xylem stem, I'm showing the picture of the grape wine. And if you look at that picture, it's a black and white picture. You will see that xylems are arranged in multiple different diameters. It's like a bike chain. I don't know if you ride a bicycle, but you know the speeds of the bike is like a smaller and then larger and then larger and then larger wheel, right? So you based on your based on the difficulty of the road conditions, you switch from one to another. Maybe lianas are doing something similar. If you look at that picture, you see one giant vessel, but next to it, there's so many other smaller vessels connected to it. Maybe just like the bicycle analogy, maybe the smaller vessels slowly help fill the next bigger vessel, and the next bigger vessel fills the other next bigger vessel, and then eventually they reach the largest vessel and then fill it up, maybe. So that's just my speculation, but that's how you build hypotheses, by making those speculations. So I think what lianas do is maybe they arrange their stem architecture accordingly. Most probably the xylem architecture, or the xylem parenchyma, which is right next to it, the living tissue. And so they control the pumping of water through aquaporins sequentially to fill the largest vessel that gets embolized. So that's my guess.
So I can continue with the other question. What was the reason for your choice of grape wine?
The reason for choice of grape wine, yes. First of all, I like Pinot Noir, and so I like wine, and Pinot Noir is the first grape genome to get sequenced, actually, and that's also one of my choices. The other thing is, if you're comparing multiple lianas with each other, with other non-liana plants, you need a yardstick, like a measurement template, and we call it orthology template. So what's orthology? Basically, you have genes, and those genes, the related counterparts of the other organism. So if you look at the evolutionary history of plants, they experience so many whole genome duplication events. And because of that, polyploidizations happened. And after polyploidizations, some plants resisted duplications and eliminated certain diploidized genes. So if you look at it, it's quite messy. The evolutionary history of plants. So when you have multiple copies of the same gene coming from an organism, but fewer copies of the same gene from a different organism, it's hard to make a comparison. So the comparison should be many to one, right? It's like a mathematical question, actually. So to solve that problem, I used grape, because grape, we all know that resisted whole genome duplications many times. It experienced only one whole genome duplication, which is the gamma, very ancient whole genome duplication. But after that, its gene sets stayed pretty much constant. So if I compare everything to grape, that will give me a very nice comparison set, because I know that the reference set that I'm looking for is in its leanest form in lianas, in grape, and also grape being grape is a liana, and so there's huge resources, genomic resources for that, because of its commercial value and historical value, and cultural value. So grape has been sequenced so many times. So there's very rich body of resources. So if I want to compare something on a metabolic pathway, for example, like the interactome, as I said, grape provides a very good resource because people made these experiments. So these are experimentally determined predictions, not predictions, experimentally determined interactions, protein-protein interactions. So all these things are already established in grape. So I was like saying, okay, grape is like the perfect organism. Why don't I use it as a reference to compare all other plants? So that's why I chose grape, and not only for personal reasons, but also biological reasons as well. So.
That's a nice answer, I think. Yeah. So our next question is, in your presentation, you stated that in almost every vascular family, there is a liana member. Mm-hmm. Is this an evolutionary consequence, or does this condition provide advantages, disadvantages in general?
Very good question. So my guess is liana way of life is very profitable, but very risky. It's like those Wall Street bets, right? So you hear all these like stock exchange questions where people are going bankrupt and some people are making a lot of money. I think Liana's strategy is very much like that. Once you hack your system to become a Liana, there's no turning back. You're a Liana, and you have to. It's in biology we call it runaway selection, like the feather of a peacock, for example. Once you're in that range, once females start to select that property, there's no turning back. You have to make it longer and longer and longer and longer. So there's probably a runaway selection in Liana way of life. So every plant family independently evolved one or two lianas because those are the ones that got into that runaway selection, let me say, and then became liana. What probably because of its risky strategy, a lot of them went extinct. We never see those. So we only do our conclusions based on what we observe. So we have to look at the surviving members of these things. So maybe a lot of lianas went extinct. We don't know. But once you get into that, past that threshold and get into the runaway selection mode, there's no turning back. So my hunch, my expectation is that lianas probably are like that. They discover a combination of genes that works great, but once you exploit it more and more, you get into a more dangerous way of life, and then you start to evolve your defenses to solve your problems in that dangerous way of life, and then you become a liana. So I think that's what it is. Probably no plants wants to be lianas. It's very risky, very high adrenaline way of living, but once you become a liana, you're a liana. There's no turning back. So I think that's what it is. So there's got to be certain checkpoints to prevent liana formation. But once you make all these checks, okay, all these transcription factors, all these genes, ding, 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 and then you've got that bingo combination, then you become a liana, I think. So anyway, next question.
Next question is, I would like to ask why co-expressed genes from lianas and co-down-regulated genes from non-lianas generate sectorizations in the gene mapping, and why do we observe these conditions on silent genes?
Yeah. So when I say large sectorization, yes, it's a separation. So we see two chunks of groups, but the separation is not entirely like 100%. So even in that set of downregulated genes, we see upregulated genes popping up, but their numbers are fewer. Maybe it's like that the symbol of yin yang. You know those the Eastern philosophies. You see this black and white, and in it there's a tiny little the opposite colors, right? So I think that's like the show of dualities in life, and I think I see a big sectorization, but inside those big sectorizations, there are tiny little pockets of the opposite category. So yes, there are big sectorizations because I think some of the central nodes in those groups have a huge how should I say, influence on other genes. So they somehow get attracted to that part of the network. So, for example, in the upregulated part, uh, I know these things because I'm staring at those on a more detailed program. For example, the most connected node, right? So it's a network. Everybody's talking to each other, think of Facebook, for example. You're a node, and you have friends, and you have friends, right? And so eventually you form a large network, and some people are very popular. They have huge connections. They have connections to people that they don't even know, right? So these things do happen because it's a strange attractant. So I think the reason why they separate it like that is because there are few nodes embedded in one part that has a huge influence, a big influence on other things. So when we look at, for example, the upregulated part, I didn't state this in my talk, but so there's a gene called PP2C, protein phosphatase 2C, and it's sitting in the center, and it has so many connections to all the other genes, even though that gene is not detected in my data set. But because the grapevine consortium has detected this gene to be influencing other genes, so when I import the data, that gene also comes and it plays a very central role, and it uh, pretty much determines the overall topology of the network, even though I don't see it directly in my data set. So I think that's the reason why certain nodes have a larger influence, and then they pull other nodes or genes with them. That's why we see that sectorization. But as I said, it's not 100% separation like the ancient symbol of yin yang. You can see the other dualities embedded in them. So, yes.
So, first of all, we have limited time,
Yes.
unfortunately. We have more questions, but yeah, I can't ask them right now.
You can send them as text. Maybe I'll try to answer them as short as possible.
We have a next professor with us.
Okay. Yes, I don't want to steal time.
So thank you so much
Thank
for your
you.
presentation. We are very happy to be with you.
This was a pleasure. You guys did a wonderful job.
And maybe after this pandemic, we can host you in our university.
Yes. First, vaccines. Haydi gençler aşıya. First vaccines.
Thank you, hocam.
Thank you, hocam.
Bye
Have a
bye.
nice day.
let us know when it's on. So, welcome to 15th annual session of AKEK Evolution Conference. This is our first session, first online session actually. So thank you for being here. Thank you for accepting to have a Q&A session. We are really sorry to not hosting you in our university, but we are really pleased the fact that you're here. Thank you.
I'm also so pleased to be here. Thank you.
So if you are ready, we can start with our questions.
Sure. I'm hearing the... Okay, sorry. My YouTube was playing. Yes, I'm ready now. Sorry for that.
Okay. So our first question is, which programming language did you prefer in animal quantitative genetics?
R. Most of the time, almost all the time, I use R because it gives me flexibility to write my own functions, especially when you work with quantitative genetics. The basic packages or packages for statistical analysis cannot be enough. Depends on your data structures. So it gives me flexibility. Yeah, I'm using R. Actually, I avoid using R at the beginning of my PhD. I was like, because I used SPSS during my master, and I insisted to use SPSS during my master at the beginning of my PhD. But my supervisor told me, you should learn R. But at that moment, I hate R because learning R was very, very difficult. But after some effort and learned it, I loved it. So I suggest everyone to learn it. Just give some time for yourself. Be patient. Just in a few months, once you get less amount of errors, you will love it.
Thank you for this answer. Our next question is, does SPP say the complexity of an organism's reproductive system? The SFP you mentioned in your presentation, similar fluid proteins.
Oh, sorry, I couldn't get the question. So, what is the question about?
So, it is, can we analyze or can we have assumptions about an organism's complexity of reproductive system by just checking the variety of the proteins?
That's a great question. So far, we don't have much information about organisms. Like we know about the few insects, human, chickens, but not a wide range of organisms. We have knowledge. We also have we know the flatworms that I work with. When I looked at the number of seminal fluid proteins among those creatures, it looks like there is a correlation between the complexity of the organism and the number of proteins. But that, for now, it's just an hypothesis. That's what I see. But definitely, will be nice to analyze for that, test for that, because yes, I expect a relation, and it will be nice to be able to test it. For that, we need more seminal fluid protein studies. Thanks for that question. That's really nice one.
Yağmur, you're muted.
Sorry. Okay. Our next question is, do we observe behaviors other than hermaphrodite creatures, such as suck behavior developed for protection from pathogens before or after mating? And could the evolutionary logic behind it be different?
Yes, there are similar behaviors in flatworms or other worms. Not always like suck behavior, but there is sperm digestion. in many worms actually, and they digest sperm. We don't know whether they really digest because of pathogens. It could be about sperm competition, or about pathogens, or to get some protein resource. But yes, there are some weird, very weird behaviors. They, for example, it's not about sperm digestion, but there are flatworms that can step themselves to fertilize themselves. Like they step their penis from their head to transfer sperm to self, actually. It's a kind of way of self-fertilization. But about sperm digestion is more about sucking and eating, I think. But yeah, there are.
So our next question is, doesn't the selection pressure of the females you mentioned increase the selection pressure even more, given the fact that the sexual election of the female is in the direction of reducing diversity? So it's actually, I think, I'm trying to summarize that. I think it's actually just putting the female sexual election in the equation. So does it cause any reducing diversity? Like in your presentation, you only actually mentioning about the sexual election of the male, but two elections together, wasn't it actually the paradox you're talking about? Like, I think that's the question.
Yeah, that's a good question too. That's actually the paradox, the question that I also tried to mention in my talk, that although females select hardly on animals, on males, for some of their traits, there is still diversity for that trait within the population, right? That's the main question. Although females choose, how still there is this diversity? And I don't know whether it is related, but in some species, of course, it is possible that not female but male choose, or both female and male choose. But usually it's about having big eggs that is expensive to produce, and having sperm cheap, which is cheap to produce. So that is why we usually see that females are the choosy one, because they have just only few eggs. Depends on the organism, of course, but they invested a lot of energy to produce these eggs. Therefore, it's usually the female who is choosing for fertilization of for male. Is that the answer of the question? Question was not very clear. I can't hear you.
Okay. We always forget that we are muted. So I think it actually explains the question, but even like in your like you stated in your presentation, this is like kind of a balanced paradox, that there's a reducing diversity, then there's election, then there is a need of there's a problem in evolution too. So I think that's a good answer.
Yeah, that's a big question still we're trying to answer. As I mentioned, genotype environment interaction, relaxation of selection. There are some like negative genetic correlations. These are some possible mechanisms to maintain diversity in the population. Just to remind.
So the other question is, how could copulator and mating plugs, which you referred to as jealous proteins, contribute to sexual selection or balance in the population outside that individual?
As I understand the question, like how mating plugs benefit to an individual, can we ask in that way?
I think it's referring to like your general presentation, you stated as jealous protein that it's actually a competitive act. Maybe it's a general question like, do competitive acts have benefits only for individual, or can we look at another larger perspective?
I think these kind of traits are, I mean, I don't know, it of course is, if the effect, the advantage is to the owner, to the individual, but evolution is, it's not individual level, it's population level. So mating plugs are evolved because probably in these organisms there is this huge sperm competition, and any kind of mutation that helps to produce some kind of mating plug should be very advantageous for that individual. Therefore, this kind of traits can increase the frequency in the population so fast, right? But in the population level, I can't say that having mating plug has an advantage for that species exactly. Yes, the advantage is about individuals, but that cause the trait is selected, right? And one more thing, maybe it supports the question that mating plug is a very good example of parallel evolution. It occurs many different animal species independently evolved actually. So it should be very advantageous.
So we cannot see any further questions in our YouTube chat box, and we would like to ask you. And we have limited time since we will have another speaker. So, is there anything you would like to add?
I'd like to thank you all a lot for this great performance. I can imagine that you're tired. It's, I think, evening in Turkey. Thanks a lot for this organization. I deeply hope that maybe next year we can meet together in Ankara. Thank you.
I was going to say that. So thank you for being with us. We are really happy to be with you. And we would really want one day to meet face to face if you come to Turkey and contact with us.
Hopefully. One more thing. Anyone can write me anytime when they have question about sexual selection, seminofluid proteins. I'm happy to answer all.
Thank you so much.
Bye.
Bye. Have a nice day.
Good. Thank you. How are you?
I'm fine.
Very excited
Yes.
to be here.
Then I will start the question and answer session. I would first start by asking, you mentioned a lot about the Bajaus and Tibetan people and Inuits in both of your talks. So could Tibetan people and Inuits have bred with other extinct species other than Denisovans that gave them evolutionary advantages as well?
With other species than Denisovans, I mean, we know that they both also, like all other people outside Africa, has a significant amount of Neanderthal DNA. And we know that humans outside Africa in general have gotten genetic variants that has to do with, for example, resistance to disease from Neanderthals. So we know that Denisovans is just one of them. It just happened to be maybe a group that was adapted to, for example, colder climates and perhaps also high altitude. But genes certainly came from other species as well. We know about Neanderthals, we know about Denisovans. And the question is, what else do we not know about? And I think particularly if you look at people in Africa, there might have been other hominins there that they have gotten DNA from. So we think now that perhaps there's a very complex story of where people in different parts of the world got DNA by integration, by interbreeding from several different species, Denisovans, Neanderthals, and probably at least one other species in Africa.
Okay. And could those gene transfers give them advantage, or were they not, didn't have any effect on them?
I mean, we know that some of the Neanderthals ones seem to have had a lot of positive selection, particularly some of the ones associated with immunity and defense. So then several different genes that seems to have a very strong effect on immune function that have been under strong selection in humans that they got from Neanderthals. The ones in Africa is more recent research, and we don't know so much about that. So that's an open question. What traits might have been conferred to humans, adaptive traits, from some of the other hominins that lived in Africa? And we don't know yet.
Okay, thank you. You can continue with the next one if you like.
Okay, there's a question. It says, could Tibetan people and Inuits have bred with other extinct species other than Denisovans that give them evolutionary advantages? You asked that? Okay. Sorry. I misread it. So there's another question. It says, what are the other physiological results of mutation in the EPAS1 gene in Tibetans? Is there any disadvantage of adapting to a high altitude environment?
So that's a very good question. So these mutations in EPAS1, they affect many different aspects of the physiology. They affect the whole cascade that happens when you don't get enough oxygen, the way your body reacts. All the different steps of that is affected by EPAS1 because it sits high up in that signaling cascade. And so there are many different physiological effects. And some of them could very well be negative. So, for example, do Tibetans have too little red blood cells in low altitude? That could be a question, for example. But that doesn't seem to be the case. It seems to be the case that EPAS1, the mutations they got from Denisovans, mostly only affect them in high altitude and not if they're in low altitude. So it seems like what the mutations have done is they have changed what we call the norm of reaction. So when we go up in high altitude, we all have a physiological response to it. It's what we call the plastic response. We start producing red blood cells at a different rate, for example. So when you have changes in humans, they can sort of occur in two ways, right? Some that are not heritable, that are just these plastic responses in response to the environment, and other ones that are genetic. In this case, the Tibetans have these genetic changes that change their plastic response. So it seems like the way that they respond to a change in altitude is different. So that doesn't seem to, I think, what we think is that there's not a negative fitness effect of having these mutations in low altitude. But of course, it raises another question, which is, in high altitude, if they're not producing so many red blood cells, then they shouldn't be as good as transporting oxygen. What are they doing instead? right? How do they function well without so many red blood cells at high altitude? And that's an open question. We don't really understand that. It's probably complex. It probably has to do with elevating the heart rate, maybe larger lung capacity, maybe some parts of the metabolism has shifted somewhat. But we don't really know for sure how they compensate in high environment through having less oxygen without having so many red blood cells.
Thank you.
Thank you. Then I will continue. As you said, there is integration between, I don't like to say species because you don't use it, that's how we use it, but there are also other incidents where the species definitions we use right now, like the mating of horses and donkeys, still exist. How does this distinction happen?
Yeah, so that's another very complex question, but we often imagine that as and there are many different models for this and many different hypotheses for this, but perhaps the most common one is that as you have population groups that become more and more distinct, in average it's disadvantageous for them to mate together, that the offspring are less fit because there are mutations that interact in a way so that they're not as fit, and that will then generate selection pressure to evolve mechanisms for not mating together. And that's, we think, why we have so many of these behaviors, for example, and also morphology that prevents interspecies matings. So an important part of the formation of species, we think, is this process where selection is actually acting to eliminate the possibility of integration, because producing offspring with the other species actually reduces the fitness, because your offspring are less well off. So I think most biologists would agree that this process is called reinforcement. It's a very important part of species formation. So the question is, when do species sort of stay connected in a network when they can exchange genes? And when does this isolation becomes complete? Because obviously it becomes complete sometimes. You know, we can't mate with a giraffe, right? I mean, there are species boundaries that are real and true. And I think that's very complex of when that, you know, when there would be complete species formation, or when actual selection might work a bit against it because the occasional exchange of genes is beneficial. So there's sort of a trade-off between having genes that might not be adapted well together, that interact in a negative way, and then also the possibility of getting adaptive variation in that can help you. And sometimes that trade-off will favor that you exchange genes, and sometimes that trade-off will favor that you don't exchange genes but become isolated.
Oh, okay. Thank you.
So I can continue.
I think
Neanderthal species are larger and motor neurons. This should have contributed to their.
Can you start again? I think you went and you think for a moment.
Okay. I can start again. Neanderthal species are said to have larger brains and more developed motor neurons. This should have contributed to their favoring by selection, but it did not. What is the reason of this? Why didn't adaptation help them?
So why did the Neanderthals go extinct? That's, of course, a very old and very interesting question, right? Why did humans spread out and took over from Neanderthals? There are many hypotheses for this. So one of them is that we require less energy to survive. So that they would need higher energy intake. And so during the Ice Age, when there were scarce resources, it might have been beneficial to have our physiology rather than Neanderthal physiology. Then there are a lot of hypotheses about behavior, that we aggressively excluded them. That's also possible. There are hypotheses about because their arms were not so good at throwing a spear, perhaps, as we are, then we were outcompeted them for hunting. So there are many different hypotheses. Now, having a bigger brain is not necessarily an advantage, in part because it uses more energy. But it's also possible that they didn't really go extinct, and that's how I see it. So if you look at how much integration there was between human and Neanderthals at the time when they integrated, maybe 50, 60,000 years ago, It's almost 10 to 1. So 10% Neanderthal DNA and 90% human DNA. Because of natural selection, we can show through time, there's become less and less and less Neanderthal DNA in humans. But originally, it was about 10% Neanderthal DNA from people outside Africa. And if you make estimates of the population density, there was about 10 times as many humans as Neanderthals. So in some sense, you could argue the Neanderthals did not go extinct. They merged with our species. We just became one species. They became absorbed into the human species. So I like to think of it that what killed the Neanderthals was not war. It was love. It was like the merging of the two species together.
Thank you.
While we're at the topic of extinction, there was a question about why do you think Denisovans went extinct?
Yeah, again, we know so little about Denisovans, it's very hard to speculate about. I mean, we can't even really speculate about the Neanderthal case, and Denisovans, we know them from just very, very, very little fossil evidence. So we know really nothing about the ecology. We know the genome, so we can guess a little bit maybe, but we basically don't know anything about their ecology or their morphology. Mm-hmm. And therefore, it's almost impossible for us to say anything about why they went extinct.
Okay, thank you.
So I can continue with another question. While the amount of blue chromosome decreases in the third slide about the recombination clock, the amount of red chromosome increases in the next generations due to recombination. Because all genetic diversity turns red over time, can we say that recombination reduces genetic diversity here?
Okay, so in that figure, the way to think about it is that you imagine some individuals that come into the population, and then they keep back breeding in with that population again and again and again. And for the first many generations, of course, the introgressed DNA that came from the other species will become more and more diluted in the population. So eventually it will stabilize at some level. So for example, now we have stabilized Neanderthal DNA in humans outside Africa is about 2 to 3%. Initially it was very high. In the first individuals for hybrids, it would be 50% by definition. A hybrid has to have 50% from the mother, 50% from the father. And now we're back down to 2 to 3%, but it's spread about everybody. So the amount of DNA that is introgressed actually doesn't necessarily go down in time, through time, except if natural selection is working against it, which we think it did for the Neanderthals. But otherwise, the total proportion in the population that's introgressed DNA would stay the same. It just becomes diluted over many people. So each person, in the beginning, each person has either 0 Neanderthal DNA or 50%. In the next generation, it would be 0 or 25%, and so on. In the end, there are very few people with 0, and all the DNA has been spread among all the people. So recombination doesn't reduce the variation. It spreads it out among individuals in the population.
Thank you.
I think, okay, this is another question about Denisovans again. It appears that some human populations gain great advantages from interbreeding with Denisovans. Could there be other populations that have similarities in their genomes to Denisovans as well? And could Bajau people have interbred with other species like Denisovans or Denisovans, just Denisovans?
So for the Denisovans, what we know about them is that people in East Asia and in South America, they have maybe 0.2% of their DNA that's Denisovan. But then Aboriginal Australians and Melanesians, so people down in Papua New Guinea, they have a much higher percentage. They have maybe 4% DNA from Denisovans. So it's a much higher percentage there. When you go to Western Europe, you might see a little bit of Denisovan DNA that's come in sort of in the northern part of Western Europe through Siberia, but in Southern Europe, really not anything. So when did we interbreed with Denisovans? We don't really know, and there are different hypotheses about it. But something seems to have happened down in Papua New Guinea, and then people in East Asia also have some DNA from Denisovans, and we think, and that's controversial, but we think that might be from a different integration event into East Asians. But it could also be from gene flow back up from Australia and Papua New Guinea up in East Asia. But we think most likely it's a different event. So there have likely been different integration events with Denisovans. We already know that there must have been different integration events for Neanderthals. It's not something that just happened once. And what there is of other species, as I mentioned before, certainly something in Africa, maybe something else outside Africa that left less of a sign than the Denisovans and the Neanderthals did.
And could there be species that have their genomes more similar to Denisovans than Homo sapiens, like Tibetan people, the other human populations that have this kind of thing?
Sorry, you're asking, are there other human populations that have something? Maybe I don't quite understand the question.
You showed a diagram that says that Tibetan people were similar to Denisovans very much, and Denisovans were like they were showing the mutation rates, I think.
Oh yeah, the phylogeny
other
Denisovans and Neanderthals are close together. And so the question is,
are there species that have something like this in their genome, like more closeness to Denisovans or Neanderthals? Then,
if it could be honest, I mean, we think maybe Denisovans in themselves were maybe perhaps quite diverse. That's kind of hard to get at from the data that we have. But there are probably many other groups, and maybe we have there's a whole continuum of different groups, and we just have defined some to be Neanderthals and other ones to be Denisovans. But maybe as we get more and more data, we'll figure out that it's much more complex than that. That there's not just Denisovans and Neanderthals, but more groups. And we certainly see within the Neanderthals also a lot of population structure, that is a lot of genetic differences between different Neanderthals. So it might be a spectrum of different genetic forms that we will be able to study in future years. Yeah.
Okay, thank you.
Okay, there's another question, but it must be answered without ethical concerns. You talked about the species formation. Can lab sport be used to help this? I mean, can designer babies be used?
To do what? For doing? Can you say the first thing of the question again? To do what with this designer babies?
I don't know. They don't specify it in the question.
But could you just read it for me again? I'm not sure I quite heard it right.
Yeah. Okay. You talked about the species formation. Can lab sport be used to help this? I mean, can designer babies be used?
For species formation, or can designer babies be used for species formation? So making new species or something. I don't quite understand the question. I'm sorry about that. I'm sorry.
Okay, it's not a problem. We just wanted to see what people asked.
Okay, yeah. No, it's fine. I think it's me. Sorry, I just didn't understand the question.
Okay, then was that the last question? It was. Is there any more? Okay, I think it is. I think that was the last question then.
Okay.
Yeah, I guess.
Well, it's been a pleasure. Yeah, some very good questions.
Thanks so much for everything you've done, for answering these and for the presentation.
Oh, it's been a pleasure. Absolutely. Thank you so much for having me do this. It's been a pleasure. All right. Have a good evening. Bye-bye.
Good evening.
Bye-bye.
Bye-bye.
konuşacak
Kim
şimdi?
konuşacak
şimdi?
Onu sordum.
Elif.
Hayır canım. Aa, peki. Okay. Hello again, everyone. We had some technical issues. We won't be meeting Professor Carlo Melli today. Thank you all for joining us here, listening to us for more than 7 hours. Thank you. That's all. Selin, do you want to add anything?
I just want to thank everyone for listening to us.
Okay.
Have a good night. See you next year, I guess.
Yes.